Okay, so let's continue with the structure of a chromosome. So when we evaluate the chromosome, there are two major parts. You have the centromere, which is the center of the structure of a chromosome. So that's right here in the center here, right? And then you have sister chromatids, which are two identical arms of a chromosome. So these sister chromatids, again, not sure why we use the feminine term, but no connotation there. But really, that we're just referring to them as sisters because they're twins. So a chromosome forms the shape of an X once the DNA has already been duplicated. So this is one actual chromosome, and this is another that's been duplicated. So they're identical and they're attached at the centromere. So we'll get more into that. So again, a chromosome diagram. So you can see here that you have an original chromosome, which would look like this. During DNA replication, that this chromosome is actually duplicated and then stays attached at the centromere in the center. So the purpose of this is, again, so that you have your identical sister chromatids attached together, because when eventually we separate those identical copies from one another to end up in two separate cells, we want to make sure that those identical pieces end up in those two separate cells. So for now, they're actually stuck together as a chromosome that looks like an X, again with the centromere in the middle, but this here is one identical sister chromatid, and here's the other identical sister chromatid. So again, this is the format that your chromosomes take after DNA has been duplicated, but before the nucleus and the cell has divided. So when we actually get to nuclear division and eventually cellular division, your DNA is going to separate at the centromere. So you end up with two identical copies of that DNA in subsequent daughter cells. So just reading through this little summary here. So again, the whole point of this process again, this is a form of asexual reproduction, or what we're going to call mitosis, is that you have an original chromosome here, and the main purpose is to make an identical copy of this parent cell, including the DNA that you have there, right? So you can see the parent cell here is identical to your two daughter cells, and that's because we duplicate the DNA. Temporarily, you have 92 chromatids, or 92 sister chromatids, but you still only have 46 chromosomes because we count the number of chromosomes by the centromere. Okay, so even though we've duplicated our DNA, you still would technically have 46 chromosomes even at this stage because we count chromosomes by the centromere. Okay, so then eventually those will separate and you get two identical daughter cells. So here's just another diagram for you from chromatin to chromosome. Right, you can see that your chromatin is more loosely organized. Again, this is when your DNA is conducting its daily activities. Your DNA actually gets duplicated before your DNA goes from chromatin to chromosome. So we'll talk a little bit more about that. But DNA is duplicated in this format, and you can see that your two identical strands are still attached to the centromere. And the purpose of that is, again, to make sure that we have a really organized division process where we make sure that those two identical copies of DNA end up in two separate cells. And then your DNA will tightly coil into chromosomes to become even more well organized for the division process. So this, again, is just a breakdown of what a chromosome is made of. So each of your 46 chromosomes has hundreds to thousands of genes, right? And every gene is made up of, obviously, our nucleotides, which are a sequence of, and this is showing you your sequence of nitrogen bases, right? And we know the order of these letters actually codes for particular proteins that your body then makes, right? So basically, each of your chromosomes hold hundreds to thousands of genes, and each of those genes, or at least the coding genes, would code for particular proteins. All right, so let's talk about, we're just going to get into an overview of the cell cycle today, and then we're going to get into mitosis and cellular division tomorrow. So what the cell cycle is basically the events that your cells go through as they grow and divide. So it's the life cycle of your cells, right? So when your cell is not dividing, it is in a period of intermission, but the things are happening there, which we call interphase. So your cells spend about 90% of their life in interphase. So interphase, if we look at this diagram here, is comprised of G1 S, and G2. Okay, so these three substages make up interphase, and you can also see we have a G0, which we'll discuss as well. But interphase is where your cells spend the majority of their lives, right? So in interphase, the cell is doing things such as growing. It's also preparing to divide for a lot of these different stages, right? They might create new organelles. They're going about their daily activities, but we do have specific things that occur in each of these substages that we'll talk about. So again, in the cell cycle, you have 4 phases total. You have your M phase, which is mitosis and cytokinesis, which only comprises about 10% of your cell's overall life cycle. I do want to make a note to you guys that I noticed in your textbooks, there is actually a typo. If you go to page 133 in your textbook, the last paragraph on the left side column that talks about the mitotic phase, the second sentence here says, it's talking about the mitotic phase, but then the second sentence says interphase accounts for only about 10%. That's incorrect. It should say the mitotic phase accounts for only 10%. So if you are doing your outline, you'll notice that there's an error there. So I just want to point that out. That interphase is 90% of the total life cycle of your cell, and mitosis or the mitotic phase is 10%. Okay, so just to be clear. Okay, so 4 phases total. So actual nuclear division and cellular division are about 10% of your cell's life cycle. And then the other time is spent in interphase, which is this G1, S, and G2. And you see that you actually have different things occurring in each. So G1, so these stand for, the G stands for gap. So G1 would be gap 1, and G2 would be gap 2. But what I would like you to think about is G stands for growth, because in both of those, we actually have cellular growth occurring. Okay, so that will help you. So again, G1 or gap 1, your cell's going about its daily activities, it's growing. This is really the earliest phase in a cell's life. In the S phase, which stands for synthesis, this is when DNA is replicated. So if you can remember, S is for synthesis. The main priority here is making a copy of DNA. And then finally, you have gap 2, which is your G2. This is when your final preparation for nuclear and cellular division is going to take place. You also have further growth that occurs and replication of organelles and stuff like that. And please remember that, again, G1, S, and G2, so these 3 steps here are all interphase. Okay, so if I look at this diagram, G1, S, and G2, the blue arrows are all interphase, and then mitosis and cytokinesis are this small portion of the cell cycle. Okay, so again, G1 stands for the first gap phase. It's the first of 4 phases. So when a new cell is born by coming from an existing cell, it enters G1. Okay, so this is the first step in any cell's life cycle. And again, this is the first step in interphase as well. So here, the cell is going to grow in size, it's going to make proteins, and it's going to do its daily job. Your S phase, so this stands for the synthesis of DNA. Again, the S phase is the synthesis phase. So this is where DNA is replicated. You'll notice that DNA is replicated before the cell divides. That's super important, that we make a copy of DNA before we actually separate the one cell into 2. So chromosomes are replicated here, or in the form of chromatin, technically. And so basically, when we say chromosomes are replicated, that means that DNA synthesis has occurred, right? So this is when we're undergoing DNA replication or synthesizing new DNA, right? Or making copies of DNA. G2, this is your second gap phase. So this is your last step in interphase. And we also call this the pre-mitotic phase because it's the step that's right before mitosis, which is nuclear division. So here you're going to have rapid cell growth as the cell is preparing to divide. And then you also get maybe additional copies of organelles that need to be copy made and stuff like that. So you're going to have final preparations before the cell divides. So these are just additional notes that I did not include in your outline notes. And again, it's going to be up to you. I'll provide the guided notes for you if you prefer that format. Or if you'd like, you can take your own notes from the notes that I post. I mean, totally up to you. Okay? But what I want you to think about are a few things. So most cells divide and thus reproduce at some point, right? Some of your cells, though, divide and reproduce a lot more often than others. There are some cells that once they reach their mature phases actually never divide. So an example of this would be your nerve cells. So we know that brain damage can be permanent, and that oftentimes, you actually can't regenerate nerves, and that's because your nerve cells are cells that typically do not divide once they've reached maturity, right? So that's why that can be dangerous. But again, most cells do divide and reproduce at some point. Maybe you can think of some of your cells that probably divide really very often, and some that would divide a little less often, right? So again, we talked about this at the beginning, but why do cells divide? For survival, because they need to stay small, so they avoid DNA overload, and they avoid cellular transport overload, right? It's also a benefit to the larger organism. So the way you grow is by cells dividing. The way you heal or repair wounds is by your cells dividing. So these are all important aspects. Okay, so we are going to talk about the M phase starting tomorrow. So I will stop here. And again, reach out if you have any questions whatsoever. And I look forward to talking with you guys.